Okay, Team Bio, welcome back to part 2 of protein synthesis and secretion. Part 1, we talked about the central dogma of biology, how DNA is read and used to make RNA, which is then read and used to form proteins. And now we're going to be talking about secretion. And secretion technically is the moving of protein extracellularly, outside of the cell, but we're going to talk about it all the way through the endomembrane system. So we're going to say movement of proteins through the endomembrane system. And the endomembrane system is all the true organelles. True organelles, hopefully you remember from class, are organelles that contain or are made up of a phospholipid bilayer. So the endomembrane system includes the nucleus, the rough and smooth endoplasmic reticulum, the Golgi body, any vacuoles, any vesicles, and the plasma membrane, as well as the mitochondrion and the chloroplast. So any organelle made up of a phospholipid bilayer would be part of the endomembrane system. Okay, so we left off last screencast talking about how ribosomes synthesize a polypeptide, the primary structure of a protein, from instructions that are encoded within the mRNA, and it does so by linking together amino acids, actually through a dehydration synthesis reaction, as it reads the mRNA message. Now, there are two different fates for any protein. One fate is that the protein is destined to live in the cytoplasm. It is going to be a protein that just stays inside of the cell and hangs out in the cytoplasm. For example, maybe this is a protein that is part of the cytoskeleton, in which case it will just stay here. So for these proteins, if they are destined to live in the cytoplasm, they stay in the cytoplasm and they are folded, or I should say cytoplasmic proteins. Cytoplasmic proteins stay, and they're folded in the cytoplasm by chaperones. Chaperones are chaperone proteins that would be also attached to this ribosome mRNA complex that would kind of be hanging off of a tail here on the ribosome and helping fold this polypeptide into its secondary, tertiary, and quaternary structures right as it's being created. Okay, so that's one possible fate for this growing protein. It's going to end up in the cytoplasm, in which case it's just folded right here by proteins that are already present in the cytoplasm. But if this protein is actually destined to be either inside another organelle or secreted outside of the cell through exocytosis, it needs to become part of the endomembrane system. So this is extracellular proteins and organelle proteins. So if it's going to end up inside an organelle or outside the cell, it needs to follow a different path. So here's what happens. I'm going to zoom out here for a second. Over on this side of the document, you see kind of a large figure, and then a little zoom in box here. This tiny little blue dot is our very own ribosome right here. So this ribosome has actually just discovered that this protein is going to end up extracellularly. It's going to end up outside the cell. So it takes this message that was in the cytoplasm and the growing protein, and it brings it over to, you know it, the rough endoplasmic reticulum, the RER. Okay, so the RER, remember, is called the rough endoplasmic reticulum because it is studded with ribosomes on the outside that are synthesizing proteins. So just to get you a little bit oriented, here is our same ribosome, here is our rough endoplasmic reticulum, this is a Golgi body, and then here finally is the plasma membrane, or the outside of the cell. And then all this blue again is the cytoplasm, and the nucleus will be somewhere up here, but we don't see it in this image anymore. Okay, we're gonna jump into this zoom-in box of the ribosome and talk about what's actually happening as a ribosome is synthesizing a protein that is going to end up inside the rough endoplasmic reticulum. Okay, so let's get right to it. Here's our ribosome. And what you can't see from this image is that the mRNA message that the ribosome was reading before, it's still here. It's still attached to the ribosome, and the ribosome is running along it. Let's say it's going in this direction. It's reading the message, and it is plucking out amino acids from the cytoplasm that are still present in the cytoplasm. So it's still doing the same business of translation, reading an mRNA message, grabbing amino acids, and linking them together to make a protein. But what's different now is that it's attached to a little pore in the rough endoplasmic reticulum, which is this view right here. Here's the RER. So it's reading the message, grabbing amino acids, but it is pushing the growing polypeptide into the space, which is called the lumen of the RER. So this is still a polypeptide. So in the lumen of the rough endoplasmic reticulum, there's going to be a bunch of other proteins that help take this polypeptide and fold it into a protein. So it's now taken on a secondary, maybe tertiary, and maybe even, well, probably just secondary or tertiary structure. So here we have a folded protein. But this folded protein is still immature. And it needs to be sent to the Golgi body in order to become mature and become a functional and active protein. So here's how it gets to the Golgi body. A piece of the rough endoplasmic reticulum pinches off and contains this immature protein inside of it. When it is completely pinched off from the rough endoplasmic reticulum, we now call this a transport vesicle. And here's where we're going to jump down to this other image here. And imagine that our protein has somehow changed color from purple to pink. So here, a piece of the rough endoplasmic reticulum has pinched off. Here we have our transport vesicle. And the transport vesicle is going to take this immature protein and bring it to the Golgi body. How it does this is that the vesicle moves through the cytoplasm. So here's the cytoplasm of the cell again. And when it reaches the Golgi body, the vesicle actually fuses with the Golgi body, kind of like two bubbles can merge to become one. this vesicle is going to become now part of the Golgi body, and in doing so, the contents of the vesicle end up within the Golgi body itself. So this protein will move through the Golgi. This is the Golgi. And in the Golgi, it is going to be modified. It might be cut in a certain place. It might have another sugar group attached to it. It might link up with another protein to become a quaternary level protein. But throughout the processing, as it moves through the Golgi, which it's going to do by like a little piece of this part of the Golgi will pinch off and then fuse with this part of the Golgi. And in doing so, this protein is going to move all the way through the Golgi body. And at the end of its journey through the Golgi, it is going to be mature and ready to be active. So from there are two different destinies for the final protein. It might end up in a transport vesicle. That fuses with the plasma membrane or the cell membrane. And if that happens, the protein that was inside the vesicle is now outside the cell. This is an example of exocytosis. When a vesicle from inside the cell fuses with the plasma membrane, and now all this protein product is extracellular, and we would say this is an extracellular protein. It is now extra, meaning outside, cellular, the cell. So this is the way that proteins that have to be outside of the cell get there. It might butt off in a transport vesicle that is destined to to stay inside the cell forever, but fuse with another organelle and become part of that organelle. So maybe this contains proteins that are necessary for the mitochondria. So this might be a vesicle that fuses with the mitochondria. And if this vesicle fuses with the mitochondria, these proteins will end up inside the mitochondria. This is a special type of vesicle that we haven't yet talked about that I should mention is this is called a lysosome. Lysosomes are like the trash and recycling center of the cell. So maybe this vesicle buds off and becomes a lysosome. It contains proteins. It's like the stomach of the cell. So anything that fuses with this lysosome is going to be broken down. So it's like a cellular digestive system. It contains proteins that break down food or old cell parts. Okay, so that's it. That's how proteins end up outside the cell. So remember early on in this year, we did a liver lab where we looked at the function of a protein called catalase, Well, catalase is an example of an extracellular protein. It starts out as a gene inside the DNA, it's transcribed into an mRNA, then it's translated by a ribosome into a polypeptide. It's then going to be brought to the rough endoplasmic reticulum where it's folded. A transport vesicle will take it to the Golgi body where it's processed and activated, and then finally a transport vesicle will bring it to the cell membrane where it will fuse, it will experience exocytosis, and the catalase protein will end up outside the cell. Catalase is present in all of our bloodstream and is there to break down hydrogen peroxide if it encounters it, which is a highly reactive compound, and turn it into something pretty innocuous, which is oxygen and water. So that would be a real-life example of an extracellular protein and how it goes from the DNA to its job outside the cell. Okay, that's all.